A very good morning to all of you, especially to our Miss Hazi. And today I'm going to talk about XNA. What is XNA, by the way? XNA is a xenonucleic acid. Xenonucleic acid, after you hear, is basically about an alien. Alien, something that we create by synthetic process, and it can replace our DNA and RNA in future. Ladies and gentlemen, let me briefly talk about XNA. XNA, in our new research, it states that it can replace our DNA and RNA in terms of all the genetic materials. Let me focus on a few things that we can able to know what is XNA. In every organism on the earth has its own genetic material in terms of DNA and RNA, but whereby humans nowadays are trying to create a new genetic material which are almost aligned with the DNA RNA in terms of XNA. So XNA is actually the X in front of the NA is determined as a Xeno. So Xeno means it's a kind of prefix that indicates one of the ingredients typically found in the building blocks that make up RNA and DNA has been replaced or almost changed completely by something different from what we find in nature that we can know as alien or unnatural. Ladies and gentlemen, now we move on to the differences between XNA and DNA. So let's focus on the differences. As we all know, the DNA composed with the nucleotides, the building blocks of DNA, are composed of 4 bases: A, G, C, and T. Attached to the bases are sugars and phosphate as their main backbone. So let's move on to the XNA. This XNA can be divided into 6 different genetic materials, and these 6 different genetic materials can be known as their own natural sugar component of DNA, with one of the 6 different polymers synthetic chemical compounds. As I say for you, the XNA can be divided into 6 components. First, HNA, second DNA, third GNA, fourth is CNA, fifth LNA, sixth or the last one is PNA. So among these 6 components, they are varies among themselves. Why? Because they are backbone. First, HNA composed of oxose as their main backbone. TNA is styrose, while the third one GNA is glycol. Fourth one is the cyclohexylene. The fifth one is ribose with extra bridge connecting the second and the fourth carbons. The last one, PNA, is desoxyribose. Thirdly, I'm going to speak about how to make this genome XNA by using synthetic methods. Ladies and gentlemen, the researchers created a synthetic genome by copying an existing one. Like for example, Mycoplasma mycoides can be transplanted into the Mycoplasma capricolum. How can we show that this mycoid is synthetic? When recreating it, the team that had already done this research added a number of non-functional watermarks to the genome, making it distinct from the wild version. Once implanted to the Mycoplasma mycoids genome, puts up the recipient cells and deleting or disrupting the 14 genes. The bacteria went on to the function normally, meaning the transplanted already functioned and worked. Ladies and gentlemen, the last one, how this XNA can benefit us? In what terms it can benefit and give some advantages to our human life? Basically, there's various of differences and various of importance to our life. Let's move on. The doctors already prescribed that these biological products, such as enzyme and antibodies, can be treated by using this XNA to certain diseases. But these drugs break down quickly in the stomach and the bloodstream. Because XNA are somewhat a kind of a foreign or a kind of unnatural thing, they are not broken down as quickly in the body as it has not evolved enzyme to digest them. Second, today's nucleic acid based medicine and diagnostic tests rely on RNA or DNA, both of which degrade quickly when exposed to an enzyme called nucleases. And XNA alternatively could be infused to those enzymes, potentially speeding the drug development process. In basically the ability to slip from the bloodstream into the disease cell means that XNA-based drugs might have a future of rival antibodies. According to nature, these XNA are replicable, evolvable, and also this could help the development of artificially genetic polymers that can bind and inhibit the proteins, such as those involved in a macular degeneration. So, as a conclusion to the audience, I would like to tell that XNA has its own benefits at the same time as its own negative impacts on human life. Just imagine, if the XNA can be projected into a human life, then means all the diseases can be corrected. At the same time, if let's say the scientists make the XNA as the main impact of human life, they can differentiate all the cells in the human body and make the human into something that they call as alien. So this will basically affect our human life. So with that, I rest my case. Thank you.